Let's talk in this video about other structures in the eye. In the previous video, we talked about the two segments, the posterior segment and the anterior segment of the eye. And we said in the posterior segment, we have the vitreous humor. We said this is like a viscous fluid that will keep the pressure of the eye and keep it open. Then in the anterior segment, we said we have the aqueous humor. Now, in the anterior segment, which is the area between the cornea and the lens, we have the iris, as we know, and we said the iris is coming from the choroid layer. So the iris technically also is separating two areas between the cornea and the lens. Those two areas are called the anterior chamber and the posterior chamber. Both of those two chambers, here and here, both are filled with the aqueous humor. However, it just the space, where is it between what and what? The anterior chamber is the area between the cornea, and the iris, whereas the posterior chamber, it's between the iris and the lens. And again, both of them are filled with the aqueous humor. So just be careful with the anterior chamber and the posterior chamber, and don't mix them with the posterior segment. Usually students get confused between the posterior chamber and the posterior segment. This is another view of both. Here at the back we have the posterior segment, whereas here we have the anterior chamber and the posterior chamber, or this was the cornea, the anterior chamber here, and this is the posterior chamber, and this is the iris. Both of them, the anterior and the posterior chamber, are part of the anterior segment. So the anterior segment, technically all of this is the anterior segment, and here behind the lens will be the posterior segment. The lens themselves are transparent layers of tissue, and their main function is to change shape to reflect light on the retina. So this is the main function of the lens. So they change shape to focus light on retina. And we said in the retina we have the photoreceptors, right? So the lens, that some students will say it's to allow the light in. That is wrong. The function of the lens is not to allow the light in, because otherwise I can remove them and the light would still comes in. No, the function of the lens is to change shape so that they would reflect or refract or focus the light on the retina. Now, those lenses, so it means that they have this ability to change their shape, and this ability of changing shape is what allows us to see clear when we are reading, like in front of us, when we are reading a book in front of us, or when we are looking far. So they will change their shape in order to reflect the light on the retina. Now, sometimes in certain conditions, we have opacities in the lens. So it means that the light cannot pass in. And that's a condition called cataracts. And in those, sometimes there are genetic factors that will contribute to this, but also like other factors might be smoking, alcohol, diabetes, age. Sometimes all of those will contribute to the cataracts. So sometimes they have to be replaced in order to allow the patient to see again. Here there is something I mentioned before, but I did not focus too much on, when we talked about the iris and the pupil, and we said those will control the amount of light coming into the eye. So in normal light, let's assume that this is a normal light. This is how the pupil is open. Remember, the pupil is the space between the iris. The iris is the colored part, right? In bright light, those muscles, there are like circular muscles and longitudinal muscles or radial muscles. So those muscles in bright light would contract or constrict, so that will close the pupil, and therefore that will limit the amount of light coming into the eye. Too much light will hurt our eyes. Whereas in dim light, circular muscles will relax and the radial muscles will contract, and therefore opening the pupil of the eye, allowing more light to come into the eye. Now, let's add some details to the neural layer or to the retina. And in the retina, we said we have the photoreceptors. However, there are more than just the photoreceptors in the retina. Now, to understand the retina, let's go back to our drawing. And now we are looking at just this small area, just this area. So I will take it and I will enlarge it and I will draw what we will see there. So from inside, so here is the inside, okay, and here is the lens. Let's assume that here is the lens. Behind this dark line, I'll talk about this dark line, we have the choroid layer, and behind the choroid layer we have the sclera. So just to make sure that we are orienting ourselves in the correct way, the first layer of the retina or of the neural tunica is something called pigmented cells or pigmented layer. So those are cells with pigments to absorb light. What does it mean to absorb light? It means they don't reflect light out. See, sometimes if you are looking at your pets or in deers at night, you will see their eyes shining, right? Because they don't have this layer in their eyes. They have additional layer that would reflect the light, and that will help them to see at night. For us, we have this pigmented layer that will absorb the light. Now, in this pigmented layer inserted to it, we will have the photoreceptors. And here we have two types of photoreceptors. We have rods and we have cones. So I'll draw them like this. So those are the rods and those are the cones. And there are significant differences between them. Rods are for dim light. Think of it like night vision. And those, they don't see colors, no color vision. But in cones are for bright light. And therefore, those are the one for colors. So during the day or even at night, if we see colors, it means our cones are active because they need a bright light. But if there is very limited amount of light, that is where the rods. And when we have very limited amount of light, we don't see colors. Okay, so those are the two big differences between rods and cones. One for bright light and for the color vision, the other one for dim light and no color vision. So what is the function of the rods and cones? Both will perform the same function, which is converting light into action potential. So they change the light into action potential, which means they get stimulated by light. And think of it like this: light will open their sodium channels and then we will have the action potential, right? So they will get sensitized or they will get stimulated or irritated or activated by light, and they will induce an action potential. That action potential will be transmitted to the second layer of cells, which is a group of cells or a group of neurons. We call them the bipolar cells or bipolar neurons. Why do we call them bipolar neurons? Because out of their cell body they have just two extensions, right? So those are the bipolar cells or bipolar neurons. Sometimes like one bipolar cell can get a signal from different rods, but for cones, each cone will send to one bipolar cell. So those are the bipolar neurons or cells. What do? They shuttle the action potential from the rods or cones into the third group of cells, which is the ganglion cells. So for the ganglion cell, we will have a cell that has dendrites. Those dendrites might connect several bipolar cells and they take the action potential. We'll talk about their axons in a bit. But those ganglion cells, usually also one ganglion cell would connect to one cone, whereas for the rods, one ganglion cell might connect to several rods. Now, the special thing about those cells is that they have long axons. And those axons will form the optic nerve. The optic nerve, what does it do is takes action potential to the vision center in the brain. So those axons will go and exit the eye through the optic nerve. So if I draw them here, those would be coming like this from all of this space to form the optic nerve. So this is the optic nerve. So now we have two things we need to be careful with. The first one is the direction of the light pathway, and then we have the signal pathway. Okay, for the light pathway, of course, we understand it's coming from outside the body, right? So it has to go through the cornea, it has to go through the lens, it has to pass through all of those layers of cells, all the way to the pigmented layer. While at pigmented layer, it will stimulate the rods and cones. Those lights will stimulate the rods and cones. So for the light pathway, we can say it's the cornea, right? Then we have the anterior segment, which contains the aqueous humor, then the lens, then the posterior segment, which has the vitreous humor, right? Then the entire neural layer, and it ends with the pigmented layer. Whereas for the signal pathway or for the action potential, it comes from the rods and cones to the bipolar cells, to the ganglion cells, to the optic nerve, and from there to the vision center of the brain. And in the vision center in the brain, that is when we analyze what we see and we understand what we are seeing. But it's really critical that you understand the difference between the signal pathway and the light pathway. Usually students will mix them. So if I ask students what is the light pathway, they will say cornea, anterior segment, lens, posterior segment. Then they will say rods and cones, bipolar, ganglion cells, optic nerve to the vision center. That is wrong. That is for the light ends at the neural layer. The light ends at the neural layer, whereas the signal or the action potential starts with the rods and cones all the way to the vision center of the brain. So just make sure that you differentiate or you are able to describe both the light pathway and the signal pathway in the correct way. Now, two more things I want to mention before I finish this video. Those are spaces that we need to be careful. Actually, one of them is a space, the other one is a region. The first one is here, the area where the optic nerve exits the eye. The area where the optic nerve exits the eye, it's called the optic disc. And this optic disc is the blind spot. So here, why do we call it a blind spot? Because there is no rods or cones in that region. So here, any light falls into this area, we don't see it. And in the lab, we will test this optic disc, or we will test our blind spot. We will try to see or to locate it in our eye. Okay, so this is one area that we need to know about. Then the other region is here, the fovea centralis. Fovea centralis is the area of high vision. The area of high vision. Here, what do we have here? We have high number of cones. If you are reading, for example, in a book, you are reading one certain word. That word that you are focusing on is reflected on the fovea centralis. So this is the area of sharp vision. This is the area of the sharp vision in our eye. Here again, the two pathways, the light direction and the signal direction. Again, the light has to pass. If we go back to here, to the entire eye, the light has to come from outside. It will cross the cornea, the anterior segment, the lens, the posterior segment, all the way to the neural layer, and from there the signal, the action potential, will be transmitted through the optic nerve. I will stop here. In the following video, we'll start talking about the physiology of the vision.